Welcome to The Explainer. Today we are diving deep into a three-dimensional puzzle that's hidden inside, well, pretty much every single cell in your body. We're talking about the incredible, intricate fold of our own DNA.
Okay, so let's just jump right in with a fact that might blow your mind. Every single one of your cells, and you've got trillions of them, contains about 6 feet of DNA. I mean, that's taller than most people.
So, how in the world is that even possible? I mean, really? Well, the answer isn't just about, cramming it all in there. It's about a super complex, beautifully organized fold, and scientists have come up with a really remarkable way to map it all out.
The technique is called Hi-C, and you can kind of think of it like taking a snapshot of the genome's social network. It basically freezes everything in place and tells us exactly which parts of the DNA are hanging out together, even if they're actually super far apart on that long, linear strand. So how does it work? Well, it's basically a three-step process. First, researchers use chemicals to freeze the DNA, which links together any segments that are touching. Then they use sequencing to figure out exactly which pairs got linked up. And finally, they use all that data to build this huge contact map. And in a really landmark study, researchers did this for both human and mouse cells to see if our folding blueprints were the same. So let's kick things off with the mouse genome. What scientists found when they mapped its network was, well, it was fascinating and honestly, completely unexpected. The first big discovery was that the mouse's network of connections isn't random at all, not even close. Instead of just a tangled mess, the map showed this highly specific centralized structure. It almost looks like a wheel with a big hub at its center. So instead of an even web of connections, the entire mouse genome seems to organize itself around two major superconnector hubs. One of them is this mysterious geneless piece of the Y chromosome. The other is a spot on chromosome 11 that acts like a magnet for the center points of all the other chromosomes. Now, get this, and this is what's really wild. Look at the sheer dominance of this one player. A single region on the mouse Y chromosome is involved in a staggering 70% of all the long-range connections they observed. It is the ultimate network hub. To put a number on it, scientists use something called a heterogeneity score. Just think of it like this. A perfectly even grid would get a low score, but a network with a few really dominant hubs gets a very high score. The mouse genome, it scored a massive 26.3, which just confirms its structure is super centralized.
Okay, so given that mouse blueprint, you'd probably expect the human genome to be organized in a similar, hub-focused way, right? Well, it's a completely different story.
And this right here just perfectly illustrates the core difference between our two species strategies. Where the mouse is centralized, the human genome is distributed. It's a network with no single king. The human contact map tells a totally different story. It is way less centralized. You can see the connections are spread much more widely across the whole genome. It looks more like a bustling city grid than that simple hub-and-spoke system we saw in the mouse. So what's the organizing principle for us then? Well, there are no dominant superhubs. Instead, it's the short, gene-rich chromosomes that are the most social. They're the ones making most of the connections, which suggests a strategy where these smaller, more active chromosomes are the ones taking the lead. And you can see it plain as day on this chart. There is a super clear inverse correlation. The shorter a chromosome is, the more contacts it tends to make. The long chromosomes are actually way less interactive. And the heterogeneity score for the human network, just 2.7. I mean, that is almost 10 times lower than the mouse's 26.3. And that just reflects a much more uniform, you might even say democratic and distributed structure. Okay, so we've got two mammals, us and mice, with two fundamentally different blueprints for organizing their DNA. So what's the final verdict here? And maybe more importantly, why does any of this even matter? So here it is, the crucial point. Evolution took two completely different paths to solve the exact same problem of how to pack DNA. The specific architectural plan for how to fold the genome is just not conserved between our two species.
This table really sums it all up. You've got a distributed network versus a hub-dominated one. Totally different key players. And get this, even the nature of the regions that are interacting is different. In us humans, these contact points are full of active genes, which is something you just don't see in the mouse. But this whole thing raises a really fascinating question. With such different strategies on the surface, did researchers find any fundamental principles that actually are conserved in both species? And the answer is yes. And here it is. In both humans and in mice, it turns out that genes that do similar jobs tend to cluster together in the nucleus. So it doesn't really matter what the overall folding pattern looks like. At the local level, function follows form. This chart shows that as the 3D distance between genes gets smaller, their functional similarity goes way, way up. This strongly suggests that there's something called transcription factories, which are like little workshops where related genes all come together in one physical spot to be activated more efficiently. So the big takeaway here is that the 3D shape of our DNA is deeply connected to how our genes actually work, with specific proteins acting like tiny architects to build these folds. But the fact that the high-level blueprint isn't universal, well, that has massive implications.
And this all leaves us with a really crucial and kind of provocative question. I mean, mice are the absolute cornerstone of biomedical research. But if the very way their genomes are organized in 3D space is fundamentally different from ours, what does that mean for how we study human diseases? This finding really challenges us to think much more deeply about the models we use and about what other secrets the genome's third dimension still has waiting for us.